Okay, so now is the time in our program we're going to transition to some work being done by the Allen Institute for Cell Science. So to kick us off with an overview of the Institute for Cell Science, here's Nathalie Gaudreault, a scientist on the microscopy team.
Hi, everyone. So I'm Nathalie, and I'm here today with my colleague Amanda, Irina, and Dave to talk to you about what we do at the Cell Science. So we study cells because they are the fundamental unit of life. We study them also because their function or dysfunction is at the core of many, if not all, human diseases. There's many effort ongoing at the moment in the cell biology world to create a more systematic, more comprehensive reference map of the cell, as well as creating some platform to elucidate the fundamental rules that govern the function and organization of the cells. So us at the Institute, okay, we are contributing to these efforts by creating a human stem cell state space. And we're hoping that by doing this, we'll answer three big questions in the field of cell biology. The first one is how gene expression and environment produce specific cellular phenotype. Basically, how we go from a linear DNA to a 3D cell. The second big question is how do we define and classify cellular phenotype? And by phenotype, we're not referring only to cell type, but all cell state. So all the different physiological conditions that undergoes in the cell. So when the cell is dividing, when the cell is stressed, when the cell is perturbed by a drug, when the cell is invaded, when the cell is migrating. The third big question is how do cells transition between different states? So that would be thinking about all the genetic material that is transferred from one cell to another, knowing that all cells come from cells, and also thinking about any epigenetic memories or post-translational morphology that can be transferred to the next cell. So this can be quite complex and challenging, thinking like this, especially when we think that there's about 20,000 genes leading to about 3 billion proteins within a cell that all form small protein complex and organize into organelles and cytoskeleton and structures to lead to this cellular phenotype or state. But we can simplify this a little bit and use two main assumptions. So the first one we're using is that cellular activities are organized and compartmentalized. So this allows us to focus a little bit more on the organelle and structures organization within the cell. And the second assumption would be that structure dictates function. And to illustrate this, I have a really good example here. Here is actin filament shown in an undifferentiated stem cell, so you can see its organization in an undifferentiated stem cell. And just next to it, you're seeing the same actin filament, but now its organization is completely different because it is in a differentiated cardiomyocyte beating, having a different function. So this big project is based on multi-scale approach, obviously, and we strongly believe that human-inducible pluripotent stem cells are ideal for this project. And Amanda will talk to you a little bit more about it in a moment. So our first big goal is to study the physical organization of the cell. So we want to combine genomics with the position, morphology, and dynamics of key structures and activities using endogenous GFP tagging and 3D live cell imaging in IPCs. We want to develop models to integrate all of these structures together and all these different state transition. And we also want to develop a platform for image visualization, interrogation, and model output. How we do this? So we have multi-team approach here. So we have 6 a very specialized team, all working together within a common goal. And to talk about the first team here, the gene editing team, we'll have Amanda today. She will come and talk to us about our legacy cell lines and plasmids. So we have at the moment up to 25 single edit stem cell lines. And we have now up to 5 dual edit cell lines. So we have 2 colors within those cells. And we also have now cardiomyocyte specific structure cell line. Amanda will talk to you about all of our protocols and methods that are used to generate these cells, these GFP tagged cell lines, and also about all the extensive genomic and cell biology quality control that are performed by the gene edited and assay development team. And she'll also walk you through our website to show you how you can get access to those cells and order them or collect more information about them. So both the stem cells, the undifferentiated cells, and the differentiated cardiomyocyte are generated for us to do live cell imaging. So this is the main thing we're doing here at the Institute. The data collection is all image-based. Irina is here today to talk to us about the work done by the assay development team and microscopy team in generating this data and also exploring a little bit that data. So very briefly, how is this data generated? So we collect Z-stack of these cells, these gene-edited cells, from a colony of cells live. These, the video is not working anymore. Okay, so what you've seen is a video. Well, it's going to play again. Okay, all these Z-stacks are collected from the bottom to the top of the cells. And in addition to the GFP tagged structure, we always use a nuclear and a cell membrane dye as a reference. We need those to then segment the cells. So here is showing you the data, the same type of data, showing you the 4 different channels we're collecting on a Z-stack. Once again, it's playing from the bottom to the top of the cells. And in addition to the cell membrane, the GFP tag structure here showing the mitochondria and the nucleus DNA label, we also collect a transmitted light. This will become very important when Dave talks a little bit later today, so keep that in mind. We've overlaid here the segmentation of these cells. And here I'm showing you 6 different examples of Z-stack collected by our team. We do this for over 1,000 replicates, and we've done this at the moment for more than 15 unique structures. So all of this is done with automation, standardization, and very stringent quality control. Those videos here are showing you to start the raw data with the nucleus and cell membrane. And then it moves quickly to the segmentation of both the structure itself and the cell membrane and nucleus. And we've left intact here the cell membrane and nucleus of only the cells that have passed all the quality control and that are moved on to the analysis. And if we take these segmented cells and we've put them all next to one another, the first thing that you can see that's quite striking is the amount of variance, the big variation between individual cells. This is true for the nucleus and cell membrane, for the cell shape, but it's also true if you look at the individual structure. And here I'm showing you 2 examples, one of microtubule and the other one of mitochondria. Now we can exploit this variance and try to make sense of it and try to draw some information from it. And here is a simple tool that we have available on our website that you can use to plot the nuclear volume over the cellular volume of all the population of cells that we've imaged so far, all the individual segmented cells. And the first thing you'll see is that we have like a nice linear relationship between cellular volume and nuclear volume for like the majority of the cell population, but we have a few outsiders here. And I'm going to let Irina tell you about those, and maybe you can guess in which state they are. Our last speaker today will be Dave. He will be representing the modeling team. And the modeling team is doing fantastic work trying to integrate all this data that we've collected. And here I'm showing you a beautiful animation that was created by the animated cell that shows you 6 different structures all at once in the same cells. So they will be talking to you why we want to integrate those all of this data together. It's obviously to see more within one cell at any given time. And finally, what I will finish with just telling you that all of our data, all of our protocol, most of our analysis, and a lot of other stories are available on our website. We invite you to visit it. You'll be lucky today to hear from Irina and Amanda walking you through some parts of it. Thank you.